Welcome to part 2 of our lecture series dealing with cell structure. The first organelle that we're going to discuss in today's lecture is known as the mitochondrion, singular, or mitochondria, plural, as it's shown on the slide. The mitochondria are known as the powerhouse of the cell. They are specifically the sites of cellular respiration. We are going to talk about cellular respiration in quite a bit of detail in some upcoming lectures, but for now it's enough to say that it is a process that involves the production of ATP, which is an energy source the cell can utilize, and that production of ATP is from the food molecules that the cell takes in. So cellular respiration is an energy converting process from which energy associated with macromolecules is extracted from those molecules and converted into a form known as ATP that the cell can utilize. The structure of the mitochondrion is a little bit different than the organelles we've talked about previously. This has a double phospholipid bilayer structure. If you look at the cartoon version on the left-hand side and compare that to the electron micrograph image on the right-hand side, the figure on the left-hand side clearly shows the double membrane system. The outer membrane is really just that, an outer membrane. It is a phospholipid bilayer. And then there is also a phospholipid bilayer that is an inner membrane. And that inner membrane has extensive folding back and forth to form structures known as cristae. This extensive folding that results in the production of cristae increases the amount of surface area of the inner membrane, and the sites of cellular respiration, that is, the molecules that are involved or the enzymes involved in cellular respiration, are mostly embedded in that inner mitochondrial membrane. So by increasing the surface area within the mitochondria, we now have much more area or location for those enzymes to be found. In addition, on this figure, you can see the region that's referred to as the matrix. That's the innermost component of the mitochondrion. And then there's also a region known as the inner membrane space. That is the space between the inner membrane and the outer membrane. And these locations will become much more important when we discuss cellular respiration in detail. The next organelle I want to discuss, I don't actually have an image or a slide for. This organelle is known as a peroxisome, and it is a membrane-bound organelle. It contains molecules that are involved in detoxification. So, for example, certain oxygen species are very reactive and hence toxic to cells. Things like superoxide or hydrogen peroxide fall into this category. These molecules, again, are extremely reactive and can cause havoc or problems or disruption to a cell. So what the peroxisome does is have enzymes involved in the detoxification of those molecules. In addition, in animal cells, the peroxisome can also be involved in the breakdown of fatty acids for fuel that can be utilized in cellular respiration. The next structural component within the cell that I want to discuss is known as the cytoskeleton. The cytoskeleton is a network of fibers that branch throughout the cytoplasm. In general, it provides mechanical support to the cell and help maintains the cell shape. It is not a membrane-bound organelle like the organelles that we've discussed previously. However, it is a component of the cell that has specialized function associated with it. In general, we can talk about the cytoskeleton having multiple functions. It plays a role in structural support, that is, maintaining the cell shape. This is especially important in animals that don't have a cell wall. And just like the name implies, the skeleton portion, just like skeletons in a vertebrate organism, provide locations for muscles to attach, provide anchorage for organs within that vertebrate organism. So within the cell, the cytoskeleton, the cellular skeleton, provides anchor points for attachment of organelles and enzymes within the cell. It is not a fixed, rigid structure. In fact, there are components of the cytoskeleton that can be broken down and dismantled and reassembled, and so it actually is a very dynamic component of the cell. This is unlike the typical vertebrate skeleton that is not dynamic, it stays in place. In addition to providing structural support, the cytoskeleton can also be involved in cell motility or movement. Changes in the cell location, so if a cell needs to move in its environment, the cytoskeleton helps with that process. In addition, there can be movement within the cell that needs to take place, and certain aspects of the cytoskeleton can play a role in that as well. Lastly, the cytoskeleton can also be involved in the regulation of biochemical activities. Enzymes can be associated with the cytoskeleton, and so enzymes that are part of a biochemical pathway, that is, steps that occur one right after the other, would make sense to have sort of an assembly line where those enzymes are lined up along the cytoskeleton. So that is another function of the general role of the cytoskeleton. So let's take a little deeper look at the cytoskeleton and break it down into its subcomponents. The first component I want to focus on are something known as microfilaments. Microfilaments are seen in the center of the slide, highlighted with the red dye, labeled microfilaments or actin filaments. In the center of the figure on the left-hand side, you can also see an electron micrograph image of those actin filaments, as well as a cartoon version of the actin filament itself. So the structure overall of the actin filament is that it is a polymer of proteins, the individual units known as actin, and hopefully you can see that in the cartoon version of the drawing overall. The actin monomer units form polymer chains, and two chains intertwine to form the complete complex actin filament. Overall, the functions of the microfilaments or actin filaments are to play a role in maintaining cell shape, resisting tension. They can help in muscle contraction, play a role in cell division, and also play a role in the movement of organelles. So back to the figure that's in the center of the slide with the red highlighted actin filaments. This is a fluorescence micrograph image of kidney cells that show those long bundles of the dense network of actin filaments within those cells. The second component of the cytoskeleton that I want to discuss is known as an intermediate filament. That's shown on the bottom of the slide in both the left-hand figure as well as the bottom right-hand corner with the fluorescence image as well. The structure overall of the intermediate filaments is composed of fibers that are wound into a thick cable. There are actually lots of different types that are based on protein composition. You may have heard of proteins known as keratins and lamins. These are definitely components of the intermediate filaments. When we discussed the nuclear structure, we also talked about the nuclear lamina. And the nuclear lamina was the network of fibers that lined the inside of the nucleus to help provide structure and support for the nucleus. The nuclear lamina is made up of intermediate filaments. In general, when we talk about the function of the intermediate filaments, they are purely structural. They are the most permanent part of the cytoskeleton. This is not dynamic, so this is not a part of the cytoskeleton that can be dismantled and rebuilt again. It is the most permanent part, again, of the cytoskeleton. Its job or role is to help resist tension and anchor proteins within the cell to different locations within the cell. Lastly, on this slide, I'm going to talk about microtubules. Microtubules are in the top portion of the left-hand figure as well as the top right figure with the electron fluorescence micrograph image. Microtubule structure are made up of alpha and beta tubulin dimers. So we have individual units known as alpha and beta tubulin. Those come together to form dimers, two units together, one alpha, one beta. And then those dimers are repeated over and over and over again to form a hollow tube or rod. So in that cartoon version in the left-hand side, you can see the dimers in the two different shades of blue, and those come together to form this hollow rod or tube. The function of the microtubule in general is to play a role in cell shape. It does help with resisting compression because of the coil-like structure of the microtubule. It can play a role in motility, in particular playing a role in specialized structures that allow for motility of a cell. These two play a role in chromosome movement and can also play a role in helping organelles and other molecules move throughout the cell. Let's look at microfilaments in a little more detail now. Microfilaments, also referred to as actin or actin filaments, tends to be the most dynamic component of the cytoskeleton. In fact, these are components of the cytoskeleton that can grow and shrink. Oftentimes, though, actin is associated with another molecule known as myosin, and myosin is a type of motor molecule that uses energy from ATP to help movement take place. So in panel A, what you're looking at is the actin-myosin interaction that occurs to allow for movement to take place. In the purple, you have the actin filament, and then in the red that looks sort of like a golf club is the myosin motor molecule. The myosin motor molecule has distinct regions, a portion that looks like the club portion of a golf club, which is also referred to as the head, and then the portion that you would hang on to or hold on to as you were swinging the golf club. When myosin attaches to actin, it does again utilize energy associated with ATP to allow for movement to take place. And I know this is a very simplified image with the arrow showing you the direction of movement, but this is how movement takes place between actin and myosin interaction. Muscle contraction definitely utilizes this actin-myosin interaction. So when we talk about muscles in a typical animal system, this is how muscle movement does in fact take place. What I do want to look at in a little more detail, though, is at the cellular level, we can look at actin-myosin interactions that allow for movement at the cellular level. In the far left in the panel B, you can see an actin-myosin interaction that was referred to as cell crawling. And in this case, what you have are interactions between actin and myosin that essentially allows for a cell to sort of creep along and move in a particular direction. What happens is these finger-like projections are established, and the actin-myosin interaction then allows for the rest of the remainder of the cell to sort of drag and catch up to where those finger-like progressions are. Then the next set of finger-like progressions would move out, and then the cell would crawl. The rest of the cell would crawl towards those fingers. In the center, you can also see actin and myosin interaction during cell division. We will talk about cell division in a lot more detail, but in animals, the way that the final partitioning of the cellular components take place during cell division is actin and myosin interaction that basically pinches the two cells apart from one another. Lastly, in the far right-hand image in panel B, you can also see actin-myosin interaction in plant cells. This is allowing for a mechanism known as cytoplasmic streaming. Plant cells have a large cell vacuole in the center of the cell, and this pushes all of the organelles to the exterior surface of the cell. What has to happen is movement needs to occur within the cytoplasm. For instance, one molecule might need to get from one location to the other, and because there's this large physical barrier in the center, how does that actually happen? Well, the cellular contents are continually moving in a circular fashion around that vacuole, and the way that that in fact takes place is due to actin-myosin interactions. Another component that I want to talk a little bit more about in the cytoskeleton are microtubules. Microtubules, just a reminder, are made up of alpha and beta tubulin dimers that repeat to form a hollow structure. Microtubules, though, organize into much more complex structures, and we're going to talk about those in just a minute. Before we talk about those structures, I want to add a few more terms that are associated with microtubules, and that is the first one being a centrosome. A centrosome is also referred to as a microtubule organizing center. It is an area that microtubules grow out of. So this is where microtubules will grow in a specific direction. The second term that you see on this slide is a centriole, and this is something that is specific to animal cells. The centriole is a region that also plays a role in organizing microtubule assembly. However, cells that have microtubules and complex assembly of microtubules can also lack a centriole. So the centriole's role is a little bit ambiguous at this particular point. Microtubules play a really important role in cell division, and they are the structures that allow for chromosome movement during cell division, specifically mitosis and meiosis. But we will come back to that concept when we discuss cell division in more detail a few lectures down the road. In addition, microtubules also play a role in allowing for movement of cells to take place. So not only do they play a role in movement of chromosomes within the cell, they can also be in specialized arrangements to form structures that help a cell move in its environment. And we're going to look at that in the next slide. Microtubules can be organized in very specialized arrangements to form structures known as cilia and flagella. These are both plural forms of the terms. Flagella is plural, flagellum is singular, cilia is plural, cilium is singular. These are specialized locomotor appendages that allow for movement of some type to take place. The flagella is a structure that looks sort of like a tail or a whip-like structure that allows for movement of a cell, such as a sperm cell in its environment as it's trying to reach the target ovum. The cilia are much shorter structures that basically look like little oars that move in a back and forth motion to allow for movement of the cell to take place, but also can play a role in just movement in general. Both cilia and flagella have common ultrastructure theme. Their organization is that there are 9 doublets of microtubules that form a ring. So again, the microtubules form this very hollow rod-like structure. Now imagine 2 of those hollow rods coming together in what's called a doublet. What you're looking at in the figure, right in the smack dab center in panel B, is a cross-section where if you counted the 2 circles that are right next to each other and went all the way around the circular structure, you would see that there are 9 doublets, and in the center there is an additional doublet. So this is the 9 plus 2 arrangement. In addition, there are other components that play a role in movement. We're going to talk about how that happens in just a minute. What I do want to talk a little bit about are the structures that you can see in panel C. Heading out from each of the 9 doublets towards the center of the structure are molecules that are known as radial spokes, and this allows for the attachment to that central component. And in the red color, you can see additional molecules known as dynein. Dynein is also a motor molecule similar to myosin that we discussed previously, and we'll look at how that works in just a minute. So what you see in the right-hand figure is a side view of one of the structures, whether it's the flagellum or the cilium, and we'll come back to that in just a minute. You can sort of go back to the figure we just looked at on the last slide and then look at this figure on the right. But what I want to focus on first is the figure that's on the left. The figure on the left has these 2 hollow rods, which represent 2 microtubules They look sort of like pipes. And those two hollow rods, one right next to the other, represents one of those doublets that we just talked about in that organization on the previous slide, and you can see them also in the right-hand figure on this slide. What I want to highlight, though, with that figure on the left-hand side is the dynein motor molecules. Again, these are in the red. They look like little feet, similar to those myosin molecules that we talked about previously. These are motor molecules, meaning that they utilize the energy associated with ATP to allow for movement to occur. So what happens is these two microtubules essentially slide past one another. If you can imagine the microtubule that's on the bottom portion of this figure staying in a fixed position, whereas the top microtubule has the ability to slide past the one I just mentioned. These motor molecules allow for that sliding to take place. So one stays fixed in position, the other slides past the first microtubule with the assistance of dynein and ATP. What that does is having one fixed in position, one being able to slide, it actually causes a bend in the structure, and that's what you're seeing in the right-hand figure overall. So this is how the movement takes place in either the flagellum or the cilium, is that you have this bending of the structure, and that's what's going to allow for the movement to occur. So the structural feature and the moving mechanism of both the flagellum and the cilium is exactly the same. The only difference between these structures is the size of the structure. The flagellum is much longer in length, whereas the cilium is much shorter in length. With the longer length, what it appears to look like is a whip-like motion of that tail-like structure known as the flagellum. But really what's happening at the base is that structure is moving back and forth with the bending mechanism that we discussed on the previous slide. In the bottom portion of the figure here, you can see that beating or bending motion that's taking place at the shorter level with a cilium. So again, the movement is exactly the same. The structure is the same. It's really just the length of the structure that differs between flagella and cilia. Most of the organelles that I've discussed in today's lecture, as well as the previous lecture, are found in eukaryotic cells in general. There were a few examples where I specified something that was specific to an animal cell, but now what I want to do is spend a little more time talking about plant cells. Plant cells are unique or different from animal cells in that they do in fact have a cell wall structure. The cell wall structure adds support and strength to the cell itself, but it is on the exterior portion of the cell membrane. So when we talked about transport, for example, in our first lecture, we discussed the fact that because plant cells have a cell wall, they won't burst when water rushes in a situation where the cell might be placed in a hypotonic type of solution. So again, plant cells do have a cell wall, which is distinct or different than what you see in an animal cell. Animal cells do not have a cell wall. The fact that plant cells have a cell wall provides some unique or interesting types of situations for aggregates of plant cells. For example, if you have this fixed rigid structure, how can cells communicate with one another if there is this physical barrier between the two cells? The answer to that is that there are tunnels or channels between neighboring cells that are known as plasmodesmata. This allows for information as well as molecules to be transferred or transported from one cell to another cell within the plant itself. Plant cells also have a number of additional organelles that are a family of plant organelles collectively known as plastids. There are structures known as amyloplasts, which store starch. There are structures known as chromoplasts, which are aggregates of pigments. But more commonly, something that you may be familiar with is a structure or an organelle known as a chloroplast. A chloroplast is the site of photosynthesis, which is an energy converting mechanism where the energy associated with light is converted into chemical energy that the plant can then utilize. The chloroplast structure is rather complex. Similar to when we talked about mitochondria, it is a double membrane system. There is an outer membrane and an inner membrane. And within the structure of the chloroplast, you have these stacks, which look similar to maybe a stack of poker chips. These stacks are referred to as grana. Singular is granum. Each individual component, each individual poker chip, if you will, in the stack of poker chips is known as a thylakoid, and it is within the thylakoid membrane that the enzymes that are playing a role in photosynthesis occur. Again, we will spend an entire lecture talking about the mechanism of photosynthesis, so we'll come back to chloroplast structure at that particular point in our lecture. The last concept or topic that I want to discuss in this lecture series dealing with cell structure is a little bit about how eukaryotic cells originated. And there are, in fact, two models for eukaryotic cell origin. They are not necessarily mutually exclusive of one another. There is evidence that suggests both may be playing an important role in how eukaryotes may have evolved over evolutionary time. The first model is something known as the autogenous membrane development model. In this model, what's proposed is that eukaryotic cells evolved by specialization of internal membranes that were derived from an ancestral prokaryotic plasma membrane. Essentially, what's proposed here is that there was some type of invagination, that is folding in of the plasma membrane, and then specialization occurring of the membrane, again, over evolutionary time. In addition, another model that's been proposed is known as the endosymbiont model. In this model, what's proposed is that certain prokaryotes lived inside larger prokaryotes. This is the endosymbiosis component. There is evidence that helps support this particular model as well, and that comes directly from looking at mitochondrial structure as well as chloroplast structure. Both mitochondria and chloroplasts are unique organelles in that they have a double membrane system that I highlighted when we discussed both of those. They are also organelles that replicate by splitting, very similar to prokaryotic cell division. The mitochondria and the chloroplasts also have a DNA component that is circular, and its structure is very much like prokaryotes. So many of the genes that are necessary for mitochondrial function or chloroplast function are found within the DNA of these organelles' own genome. They are referred to as the mitochondrial genome or the chloroplast genome. In addition, both mitochondria and chloroplasts have their own ribosomes. These ribosomes are structurally distinct from the ribosomes in the cell of in the cytoplasm of the cell in question. These ribosomes are much more similar to prokaryotic ribosomes than the eukaryotic ribosomes, again, found in those cells proper. So these lines of evidence suggest that there is perhaps this endosymbiont model, again, combined with the autogenous membrane development model, may have allowed for the evolution of eukaryotic cells. There is fossil record that suggests that the oldest eukaryotic fossil is somewhere around 1.5 billion years old.